Hi guys, I'm here to talk to you about how to do Punnett squares. I just want to give you a little background information first before we start, and the first thing you need to know is what an allele is. You're going to have to forgive my terrible handwriting on this iPad. The first thing is an allele is a copy, an alternate copy of a gene. So you know that you have these things called genes that control a lot of things about your traits. So for each trait, you have one or more genes. So let's just start with a simple situation where you'd have one gene for one trait. So if you have two copies of your chromosomes, that's what these are, okay? This is a diploid person or organism. If this is you, maybe this is your chromosome number 1, and maybe you've got a gene here at this location, okay? And you've got two copies of this gene. So maybe one copy would be a dominant copy, and maybe one copy would be a recessive copy. And that's your genotype for that trait, and this would yield some sort of phenotype. So if this was a pea plant, maybe, you would have a dominant capital T for tall and a recessive lowercase t for short. But since capital T shows us that we have a dominant gene, this is going to yield a phenotype of tall. Okay, so that's the phenotype. The genotype is this right here, this little doodad. This set of letters represents the genotype. That's the actual genes that this organism is carrying. And there are two copies because there are two sets of chromosome number 1. Alright, so if you can understand that basic concept, then we can talk about doing a Punnett square. So let's say we have one parent who has two genes for tall, two alleles for tall rather. Okay, and another parent is heterozygous. We've got a homozygous dominant individual and a heterozygous individual, and they have some offspring. And that offspring, what will that offspring look like? So we need to do a Punnett square. This is a way to determine the probability that the offspring will look a certain way. So here's parent number 1, that's this parent, okay? And then parent number 2 will be on the side. I always do the first parent at the top, the second parent at the side. That way I don't get confused. So then you just bring the T's from the top down and from the left over. And then I'll take this top T right here again and I'll bring it down. And I'll bring this T and I'll bring it over to the side. Do the same thing on the right, top down, left to the right, and then the capital T and the lowercase t. So what this is showing us now is that these two parents right here, their offspring have a 25% chance of having this genotype, two capital letter T's, which is homozygous dominant, and then 50% chance, oh, I just said that wrong. I'm sorry, go backwards. Thought I was doing something else. Okay, back up just a little bit. Okay, they have a 50% chance of being homozygous dominant and a 50% chance of being heterozygous, because I've got 2 out of 4 that are homozygous dominant and 2 out of 4 that are heterozygous. Okay, so let's do another example. Another example, parent 1 is, let's say the parent 1 is a heterozygous individual, and parent 2 is also heterozygous. Okay, you draw a Punnett square, bring parent 1 here, and parent 2 here. Now we fill in the blanks. I'm going to give you a second to do it on your own, think about it, and then I'm going to start filling it in. Parent 1 is heterozygous and parent 2 is also heterozygous. So we bring our capital T's, and then we always put the capital letter first. So even if you would normally want to put the other letter first, we put the capital letter at the beginning. So for example, this would not be a valid genotype. We don't like that. Okay, we always put the capital letter first. Alright, so our resulting offspring then are going to show us that 25% of them have this genotype, heterozygous dominant, 50%, 2 out of 4, have the heterozygous. Did I say hetero? This is homozygous, this is heterozygous. Homozygous. And this is heterozygous. And what this word really means is same within the zygote. Same in the egg, or same in the new individual. Remember, a zygote is a fertilized egg. Let's write that down. Zygote, fertilized egg. And remember that a fertilized egg is a new individual. It's the offspring of the two parents. Okay, so back to the Punnett square. 25% then are homozygous dominant, 50% heterozygous, and then 25% are the recessive. Okay, and the recessive one is also homozygous, but you're not often going to hear anyone say homozygous recessive, because there's only one choice. If you have a recessive genotype, it has to be the little t. And let's talk about their phenotypes. If this allele is for either short or tall, okay, the genotype big T gives us tall, and the genotype big T little t also gives us tall. So that means that 3 out of these 4 are tall. Okay, that would be 75%. And then that also tells us that 1 out of 4 would be short, and that's 25%. Now what this does not mean, it does not mean that out of 4 kids, 1 would be short and 3 would be tall. What it means is for each offspring, for each individual offspring, has a 75% chance of being tall and a 25% chance of being short. So these 2 individuals could have many offspring that were all tall and never have an offspring that's short, but each one has an individual chance, 25% chance of being short, if that makes sense. Sort of like playing the lottery. If you play the lottery over and over and over again, you don't increase your chances of winning. It just means you have the same chance of winning each time. You don't get luckier each time you decide to play the lottery. Okay, so that's a basic tutorial on how to do Punnett squares. If you have any questions, come see me. Otherwise, we'll see you with the next video.